Hey, welcome everybody to our second special session of the forum, where we'll get to hear all about some amazing work being done to detect and control invasive Phragmites. I'll pass it over to the moderator, Alex, who is a program development coordinator at the Invasive Species Center.
All right. Thank you, Maddie. And thank you for joining us for our special session on Phragmites, detecting and controlling Phragmites. To start off, we'll get Darissa Vincentini from the Invasive Species Center to talk about the Ontario Phragmites Action Program and give an overview of that.
Great, thanks, Alex. Can you just confirm you can see my screen?
Yes.
Awesome, thank you. All right, good morning, everyone. My name is Darissa Vincentini. I'm the Northern Ontario Regional Coordinator with the Invasive Species Center for the Ontario Phragmites Action Program. This morning, I'm just going to give a brief overview of the program, kind of introduce the session before we hear from some great practitioners and managers and researchers in the field on Phragmites. You'll often hear me refer to the Ontario Phragmites Action Program as OPA for short throughout the presentation. And yeah, we'll just get started. So firstly, a bit of background. Invasive Phragmites is often considered Canada's worst invasive plant. It's an aggressive grass that spreads very quickly, causing a number of negative impacts on the landscape. This includes the impacts to the environment by creating monocultures, pushing out native species, impacting species at risk, and altering water flows. Social impacts like increasing risks of wildfire, reducing sight lines along roadways, and limiting access to shorelines and recreational activities, as well as significant economic impacts, including damage to infrastructure, impeding drainage and increasing flood risk, decreasing property values, and so much more. So in 2024, the Ontario Phragmites Action Program was born with the support of Ministry Natural Resources to help manage and reduce some of these impacts. The OPA program, now entering its third year, is jointly delivered by the Invasive Species Center and the Nature Conservancy of Canada with the help of numerous delivery partners working together to reduce the impacts of invasive Phragmites across Ontario. The program expands on existing efforts across the province by supporting the shift to a landscape-scale collaborative approach to invasive Phragmites management through coordination across jurisdictions. Through the program, we offer funding opportunities, management guidance, DNA screening kits, training opportunities, and support the development of Phragmites management area working groups. Phragmites, like all invasive species, do not respect property lines. So through OPA, we are working to solve the commonly encountered issue you see in this photo. One property, in this case, the road allowance is being managed. So over here, you can see some dead stems of Phrag is being managed and treated, while the population that extends beyond that property onto another is being left unmanaged. We know that this is ineffective and results in ongoing and costly management. So OPA brings rights holders, municipalities, conservation authorities, Indigenous communities, NGOs, and industry partners together. Basically, anyone who has a role to play in Phragmites management or prevention together at a table to discuss strategic, regional priorities and take collaborative and coordinated action. The program supports practical solutions to tackling issues like this one. So the way that it's kind of rolled out on the ground, Ontario is split into 4 regions. We have the Eastern, Southwestern, Central, North, and North region, each with a regional coordinator, so myself for the North, who serves as a link between the broader provincial program and the local groups on the ground. The regional coordinator supports through additional training, genetic testing services, the Invasive Phragmites Control Fund guidance, biological control once available, identifying gaps and patterns on the landscape that impede effective management, and connection to large landscape scale land users and owners such as MTO, hydro, and rail lines. So if you're interested in getting started on Phragmites management, or you want to know who else is working on it in your area, you can get in touch with your regional coordinator for your region. And then within each region, we have Phragmites management areas, or what we call PMAs, which are spatial units where Phragmites monitoring and control activities are coordinated by a working group. The working group is a collection of organizations, individuals, business and industry, and government representatives and other key implementation partners who contribute to a local implementation of our Phragmites regional guidebook. We currently have just shy of about 30 of these developed so far across Ontario. They typically meet about 4 times a year to discuss priorities, division of labor, funding, and other key project planning pieces to ensure a more kind of fulsome approach to management within the area. One of the program supports we offer is the DNA screening program, which helps increase the confidence in practitioners as they identify and subsequently manage Phragmites on the landscape. The two subspecies can be difficult to field ID as their morphological characteristics can vary, especially in different parts of the province. So in the North region, for instance, native Phragmites is much more prevalent, and the invasive Phragmites is a little bit more stunted. So you don't typically see the massively tall stands that you would see along the shores of Lake Erie or Lake Huron in Southern Ontario, for example. More on morphological characteristics, you'll hear from Dr. Michael McTavish and even Ian Jones later, so I won't get too much into that. But to make things a little bit more challenging, the invasive and native subspecies have been found to hybridize. So although still seemingly rare, there have been 5 confirmed hybrid populations in Ontario so far through this program. Hybridization increases the likelihood that invasive Phragmites can go unnoticed on the landscape by having the potential to present itself as native, but still pose invasive impacts. The results of this program highlighted that 16% of field IDs from practitioners that sent in kits were misidentified, demonstrating the need for this program to support Phragmites management. Another key pillar to the Ontario Phragmites Action Program is the Invasive Phragmites Control Fund, which provides grants to organizations to support on-the-ground action, including Phragmites mapping, monitoring, and planning, Phragmites management, and management innovation. You may have caught my colleague Chelsea yesterday morning in the grant special session, who provided an excellent overview of our granting program, which was followed by presentations by some of our successful grant recipients. So I'm not going to get too much more into detail here today, but if you missed the presentations yesterday, you can catch the recordings on our YouTube channel. We also offer a number of resources to support on-the-ground action. We have the Regional Plan Guidebook, which supports the development of Phragmites management areas, offering a step-by-step guide to make it as easy as possible to pick up and implement. We have the Best Management Practice Guide which supports invasive Phragmites management decisions, providing an outline of different management techniques and the effectiveness of each. The Clean Equipment Protocol for industry practitioners to support reducing pathways of spread of Phragmites across the landscape. And we also have other fact sheets like the cost-benefit analysis outlining economic impacts of Phragmites and many others. So if you're interested in any resources, you can also reach out. So again, just to recap some of the ways that OPA can help support some of the Phragmites work that you may be doing on the landscape. We offer map creation support, genetic testing program, biological control program once it's available. We also have some upcoming online training, outreach and education support, invasive Phragmites control fund, and as well as resource review and creation. And I'll leave it at that for now. You can please visit our website for more information. If there's questions, I can type the answers into the chat as well. And you can always reach out afterwards. Thank you so much.
Awesome, thanks, Teresa. That was a good overview.
Thank you.
Next up, we will have, my display is glitching out. There we go. Next, we'll have Jay Dertinger from Shawanaga First Nation, and they'll be talking about protecting habitats and resources for all generations. And just a reminder to everyone before Jay gets started, if you could put any of your questions in the Q&A box rather than the chat. And with that, I'll hand it over to Jay.
Thanks, Alex. I'm going to start sharing my screen here. Can I get a thumbs up just to make sure everyone can hear me okay? Awesome. Okay, so Ani, hello. I'm Jay. I've been the head species at risk technician for Shawanaga First Nation Species at Risk program for 3 years now. So I've been helping lead efforts to conserve wildlife and habitats and helping lead projects focusing on a variety of species at risk, as well as habitat restoration programs like this one. So I'll be talking about our FRAG program today. So Shawanaga is a small community along the eastern coast of Georgian Bay. We have 3 different main parcels we do work in the lands department, which would be the main reserve in the bottom right corner of the map there, and then the landing, and also Naskutang, which is sort of the diamond shaped one near the top, and also the Shawanaga Island IPCA that is highlighted in green there with the green outline. But the range of Shawanaga's traditional territory is actually much larger. As you can see on the top right, spans sort of all the way to the Quebec border. So Shawanaga is home to a really diverse, it's a really diverse area for wildlife, many of which the species that are here are reptiles and amphibians. And a lot of them are species at risk reptiles and amphibians. So that's kind of where the program started. It focuses on indigenous-led conservation and stewardship, which looks like building community capacity and ensuring our healthy ecosystems are maintained into the future. So the program began with a lot of reptile work, but expanded into fish work like sturgeon monitoring, which we just started doing last year. It was really exciting in the rivers leading into the bay, and then also the Bats of Georgian Bay initiative, which was a multi-year initiative that spanned over 10 different First Nations along the eastern coast of Georgian Bay and the Bruce Peninsula, which was also a really exciting program. So considering we're at an invasive species forum, I'm assuming a lot of you are familiar with our usual suspects here. We have the invasive subspecies of Phragmites that's so prevalent in Ontario, and also purple loosestrife, but most of the focus will be today on the Phragmites removal and management. So here's a map of all of our survey sites mapped through Survey 123. So we've got a number of invasive sites that are mostly along the highway, and then also a number of native Phragmites sites, and which we've been especially trying to map more and more in the last year or two. So they're pretty prevalent up as you get up the 529 towards Naskutang. You can see all the blue dots up there, and then some on Shawanaga Island and a couple on the landing. And we've also sent some for genetic analysis as well to the Invasive Species Center, and some of the patches at the landing that we weren't sure whether they were native or invasive, because they kind of popped up as the water levels changed on the bay, were actually found to be native. So we're looking forward to sending some more for genetic analysis, and we have some really dense native patches that exist out on the island and in Naskut that we're not sure if they're native or hybrids, just because of the way they grow, but like from ID characteristics, they seem to be native. So remains to be seen what those will turn out to be. So for our management techniques, we are actually requested by the community not to, or to omit the use of chemicals in our removal. So we mainly use techniques taught to us by Georgian Bay Forever, which is one of our main partners in the FRAG project. And so we use cane cutters for aquatic patches mainly, with a cut to drown technique, which requires cutting the plant below the water surface and then revisiting sites to manage regrowth, so it drowns out the stalks. And then for the harder terrestrial sites, these are a lot more difficult to manage. We use root cutter spades, sometimes regular spades, but we have special root cutter spades. So basically, you want to cut an inch to a couple inches below the ground, but try not to disturb the rhizomes or the soil around it. And then again, you have to come back to manage regrowth. And so we cut and bag seed heads as well and use hedge trimmers for larger patches, especially with a lot of old growth. So once the seed heads are removed, go through with the hedge trimmers and cut those down, and then everything gets left on a dry spot to rot in the sun, or it gets bagged and then rots in the bags and then goes to landfill. So basically, this project started with funding from WCPP and the IPCF programs. We're very thankful for that to help us get off the ground. And basically, the whole point is to ensure healthy wetlands for species at risk, medicinal plants, and other culturally important species that are going to be using the wetlands. We wanted to address invasive species through a couple different means. So one is building capacity in the community. So training with community members, staff, neighboring First Nations, building capacity through access to like the proper tools. So purchasing all the equipment we needed, and then also lending out that equipment and lending our time to our neighbor First Nations and partners to make sure everyone can participate in the management of invasive species. And we've been building a network of regional partners and also increasing our knowledge and understanding of the current spread and abundance of invasives. So initially the program, we went through a couple iterations of names. We had the ISDF, and we've recently settled on protecting habitats and resources for all generations. We just thought it was clever, and also, just better conveyed our general mission of protection rather than, destruction. So we started with a couple main objectives, mapping all invasive patches along roadways within about 20 kilometers of Shawanaga. We wanted to start to map interior wetlands as well to understand the spread throughout the reserve. We wanted to cut and manage around 10 sites and work with 1 to 2 partners a year to manage their sites. And so we've come a long way since the start of the project. We now have over 65 sites mapped from Survey 123 alone, over 45 managed, and spanning our scope now spans across about 85 kilometers of roadways. We've revisited over 20 previous sites for active management. And there's also been over 25 sites managed by partners. Shout out to the Ministry of Transportation and Wasauksing First Nation for helping some of the highway patches and then helping us with some other patches. And we've also managed to create a network of 8 total partners. So for this project, we've been partnering with neighboring First Nations, townships, and local conservation groups especially. They've all been instrumental in helping get this project off the ground and making sure it moves forward in a good way. And so now that you have a general overview of the project, I kind of want to move into sort of a case study of one of our sites that we've been managing for since the very beginning of the project. So this is at Skerryvore Road, which is just off of the main res parcel, but is in the traditional territory of Shawanaga and is also generally in our working area for a lot of our species at risk projects within our buffer. So the first picture here is this dense patch that was reported on iNat in iNaturalist in 2019. So as you can see, there's not a lot of seed heads yet. It's like pretty fresh growth. And then it was reported again in 2021 near the start of our program. And as you can see, there's a lot of seed heads coming through here, some older growth, some more mature growth. And then so we started managing it in 2022. I don't have any pictures in 2022 pre-management, but this is pre-treatment from 2023, where we came in and got rid of the rest of the patch for the first time. So as you can see, it was like it's extremely dense before it was partially knocked down and cut. And then here's pre-treatment, and then that same day post-treatment. So we piled it up to rot in a dry spot on the patch. The only other really area would have been on the road, which is we can't really do that, and it was too much to bring out. So we chose to let it rot. So yeah, this is the year that most of the management was done at the spot. As you can see, there's like pretty much no standing water at all. The only species in here besides Phragmites was a couple patches of spotted jewelweed around the edges. And so we cut this one with the hedge trimmers and cane cutters and spades, just a combination of things, and then piled it up. And then, as you can see here, there was actually minimal regrowth of Phragmites. This is pre-treatment in 2024 in July. So the only thing that really grew back was along the back corner, and we actually had a return of quite a bit of standing water when there was absolutely none before, which was really cool to see. Still, the only species we really saw pop up in abundance was the spotted jewelweed, but at least that's, better than the Phragmites that was there. So we were happy to see that. And then, oh yeah, this is post-treatment. You can't really see even that much of a difference. It was mostly just kind of towards the back. And then we have this past season where we went in for a treatment, and to our surprise, we actually had a bit of a biodiversity boom at this site. So we had a number of plant species returning. We had insects and vertebrates now using the patch readily. And we sort of saw a stream start forming and like meandering through the patch. This is just kind of a summary of what I just talked about, so I don't really need to go through that again, but we managed it for about 4 years, and then yeah, on this last year of management, we really saw just like a boom in all the native plants coming back. So here's just a quick overview of the species diversity that we saw return to this patch. We had over 16 native plants, many of which, almost all of them, were wetland indicator species. We had over 4 native insects. I'm sure if we did a formal insect survey, we would have come up with a lot more, and then a few other introduced species that still existed, mainly on the roadside or on the bank near the road that need to be removed as well. But here are just some pictures of the biodiversity we saw just in like one afternoon of INATing at this spot, which was really fun and exciting. So you can take a look at that. We found a couple sedges and some grasses and a number of other wetland species, which was super exciting. We had grasshoppers mating and a ton of green frogs as well. So yeah, just seeing the regrowth and return of biodiversity at this one site over the course of a couple years has been really exciting, and just like a testament to what needs to be done in terms of like capacity growth and also continued management over time. And that's kind of the whole goal of our program. So it was really cool to see. So next steps with our program, we want to test prescribed burning as a potential management technique in collaboration with the Shawanaga Fire Department. That's kind of next on the docket. Continue our goals of building capacity with our partner First Nations. Like last year, we helped Wasauksing First Nation with a couple pretty large patches, and we want to continue to do that every year as much as we can. Monitor our patches long-term to prevent regrowth and further spread. Potentially collecting and spreading native seed for wetland plants to help speed up restoration efforts. And use ELC and other habitat delineation protocols to properly classify these habitats, and then redelineate them years down the road to basically monitor changes in the habitat types would be something else that we want to look into. And I think that's all I have for you now. Miigwech to the Invasive Species Center for hosting and having me. And I will take some questions if I have time. But otherwise, you can find our work at ShawanagasPeciesAtRisk.ca. I forgot to put a link there, but I can put it in the comments. And yeah, thank you again, Miigwech.
Awesome, thank you, Jay. That was really interesting. Really great seeing all of the native species growing back after management. That's really cool. We do have a few questions here. How come you take the seed heads to landfill instead of burning them?
Like I said, we haven't tested burning as like a practice yet. We don't necessarily have like an area where we can safely do that. So that's going to be something we want to do in the future.
Great. How effective and efficient is do you find manual frag management, especially for larger colonies?
Yeah, so it can be pretty time intensive and labor intensive. It requires, I mean, a lot of staffing, a lot of volunteer work, and that's also why we, for the larger patches, we tend to start at least with some of the hedge trimmers, because it just makes it more reasonable to manage. But yeah, it is labor intensive. It requires, years of revisiting the site and, continuing to plug away at it. But I mean, we have seen, as you saw in the presentation, we have seen a lot of, native regrowth at sites that we continue to put that effort into. So I think it's definitely well worth it for areas that maybe communities don't want that chemical removal and stuff like that.
That actually kind of leads into the next question. I am in a similar situation where I'm unable to use herbicide to treat frag. Have you considered any additional manual methods, for example, smothering with sheets after you cut?
Yeah, we actually have done that at a couple sites. We have some pretty massive thick tarps that we've used. I mean, even in the one that I showed as an example, that one was initially like tarped with like a giant, really thick tarp. And we didn't see like any regrowth from the area that we smothered with that. And then we eventually removed it and just like left it to completely rot in the sun. But yeah, so we have seen some success with both of those methods, just leaving it out and also tarping it.
Have you found? Oh, I lost the question. There. Have you found cut to drown to be effective? It seemed they seem to have used cut to drown and didn't see much success. So if there are any suggestions you may have with regard to that management practice.
Yeah, so we haven't quite yet seen full recovery from our cut to drown sites. So maybe they take longer, or maybe like I'm not sure if the seed bed is just full of frag in those spots, but a lot of our sites that we've seen more success with controlling our terrestrial. I mean, like the regrowth is still not as dense as it once was in some of these cut to drown sites, but it's kind of a work in progress. But if you did want to ask someone more about that, I would contact Georgian Bay Forever. Their scope is a lot larger for aquatic Phragmites removal, especially, I mean, their work area specifically all across Georgian Bay. So I would definitely reach out to them, and I can get you in contact if you want to as well.
Okay, awesome. Thank you. That's all the time that we have right now for the Q&A. So if you want to stick around and maybe answer some of the questions in chat, that'd be awesome. Yeah, thank you so much for for your talk and the answers to those questions.
Thanks, Alex.
Next up, we have Ian Jones from the University of Toronto, and he'll be talking about accelerating the implementation of Phragmites biocontrol.
Thank you, Alex. Shall I go ahead and share?
Yeah, you're good to go.
Okay. We should be good to go there. Can you give me a thumbs up if that looks good? Okay, perfect. Thanks, Alex. So yeah, hi everyone. I'm Ian Jones. I'm one of the scientists that leads the biological control program for invasive Phragmites. So that's what I'm going to talk about today, and particularly I'm going to talk about the process of kind of accelerating the program and moving beyond a purely research phase of the program and into more of a kind of research and integrated and implementation phase. So just to give you a quick overview, I'll talk a little bit first of all about what classical biological control is, and importantly, how we make it safe in modern biological control. I'll give a brief update on the program status up to this point, but then I'll talk in a little bit more detail about some work we've been doing, developing methods for moving insects from sites where we already have these biological control agents established on the landscape, and we're calling those nurse sites. So our ability to move insects from nurse sites into new locations, because if we can develop these methods, we can rely a lot less on lab-reared insects, and we can produce and we can conduct far more releases each year. So I'll finish off by talking about some of the ways in which we plan to use those new techniques to expand the program across the province. So first of all, what is classical biological control? Well, Phragmites, for example, grows as a native plant in Europe. And as you might expect, it plays a lot more nicely with other plants and animals in that part of the world, in part because it's held back in its growth by its interactions with herbivores and natural enemies that it's co-evolved with. But when Phragmites was introduced to North America, it was introduced in the absence of any of those herbivores. And so it's partly for that reason that the Phragmites just grows in this kind of unchecked, hyper-competitive way. And that's where we see this kind of invasive growth habit. So through classical biological control, what we aim to do is reunite Phragmites with some of those natural enemies and herbivores and knock the Phragmites back enough to allow native plants an opportunity to have a foothold back in these environments. But that's an important point to raise about biological control generally, which is that the end goal of biological control is not the eradication of the target weed. So we're not looking to eradicate Phragmites here. We're just looking to knock it back in its competitive ability to the extent that native plants and other organisms can start to kind of return to these environments. And that's part of the reason why biological control works. It's one of the benefits is that it works very slowly. It doesn't create kind of a big disturbance event that can be potentially pounced on by other invasive plants. It just happens very slowly, and hopefully we'll start to see these native plants starting to return. But how do we make biological control safe? I mean, this is an obvious question for everyone. And how have we moved away from ecological disasters of the past, like the introduction of cane toads to Australia in the 1930s, to modern day 20th century success stories like the control of purple loosestrife and St. John's wort? And the main answer to that question of what we've learned is the importance of host specificity in our biological control agents. So when we began searching for candidate biological control agents for Phragmites, this was happening in Switzerland in the late 90s, and we were looking only for insect biological control agents that we believed to be highly host specific to Phragmites. And with that in mind, two insects rose to the top of that list quite quickly, and that's Arcanara neurica and Lanisa geminopuncta, both stem-boring moths. So despite our initial belief that these insects were highly host specific, these insects were then put through a long and rigorous process of host range testing. So host range testing really is the foundation of modern biological control. And it's why in the world of weed biological control, there have been very, very few cases of non-target effects of biological control agents since the 1960s or so, certainly that wouldn't have been predicted by host range testing like this. So the way it works is that we will generate a long list of test plants, starting with the plants most closely related to the target weed, and then moving out in terms of relatedness to incorporate plants of economic or cultural significance. With each of those plants, we're conducting tests to see whether our candidate biological control agents will feed on those plants at all, whether they can complete any part of their life cycle on those plants, will they lay eggs on those plants. And it's only when we're totally happy that these candidate insects are highly host specific that we will petition to release them. And as I mentioned, this process for Arcanara neurica and Lanisa gemina puncta took 11 years. So it's a long and rigorous process. The way those experiments work is they often start with very simplistic no-choice tests, where the insects will be in a container with the test plant. And we're asking the question, will the insect feed on those plants at all, or will they just die? Then the experiments become more and more complicated, looking at whether any of these insects can complete their life cycle on any of these plants. And eventually we'll be conducting field studies that much more closely mimic the kinds of experiments or the kinds of environments that we might be releasing these insects into. Unfortunately, in 2019, we had petitioned to release these insects, and that petition was approved, and so releases began in 2019 in southern Ontario. So since that time, we've released about 40,000 insects across 60 sites, and we continue to monitor those sites, and we happily have seen in the vast majority of those sites that these insects persist. They're reproducing successfully, they're overwintering successfully in these release sites. And we also check during those monitoring periods for any potential non-target effects. And I'm pleased to say that we're not seeing any non-target effects at all. So some of our first releases were conducted upwards of 5 years ago now. And at some of those sites, we're now starting to see obvious visual differences in the vigor of the Phragmites. So this particular site is on Rare Charitable Reserve, where the releases were actually conducted by Claire Shon and Rebecca Rooney of the University of Waterloo, where they're doing work primarily looking at the potential return of native plants to these sites. But it's one of our best sites in terms of the visual difference that we can see as a result of the release of these biological control agents. So all of this is very, very encouraging for the program, and it's been achieved entirely through the release of lab-reared insects. So we rear the insects in the lab at the University of Toronto. The eggs are the overwintering life stage, and so in the spring, we either release them as eggs by placing them in these plastic release containers that you can see on the top of the slide there. When the eggs hatch, the larvae will drop through the mesh at the bottom of those containers and spread through the Phragmites from there. The alternative is to release these insects as larvae. So in that case, we allow the eggs to hatch in the lab, and we use a paintbrush, a fine paintbrush, to individually inoculate a single first instar larva into cut Phragmites stems. And then those stems are then released in these florist foam blocks. So those larval releases are, by a distance, our most effective release method. But as you can imagine, they're fairly labor intensive. And so there's a limit to the number of those releases that we can conduct each year. And so a big part of the program now is developing ways that we can conduct more releases, slightly less labor intensive releases. But based on the work that we've done so far, I showed you those 60 release sites that we have across the province. And a good number of those now, we have large, robust and kind of persistent populations of these biological control agents that are existent on the landscape. And so we'll be monitoring those, what we call nurse sites, to look at dispersal behavior of the insects, so the natural spread of the insects across the landscape. But we also want to give that process a helping hand so that we're able to actually collect insects from those nurse sites and move them into new locations. Because if we can do that, we can massively increase the number of Phragmites infestations that we can inoculate each year. So these insects, both of the agents are univoltine, so they have one generation per year. The adults fly in mid to late summer and they lay their eggs underneath the leaf sheaths of the Phragmites stems. So as I mentioned, those eggs are the overwintering life stage. So the eggs will remain under those leaf sheaths throughout the fall and the winter. And so to us, the most obvious way to harvest insects from nurse sites was to collect Phragmites stems from those sites in the fall, in the hope that we're collecting eggs that are on those or under those leaf sheaths. So in the summer of 2023, for the first time, we started to test this out. We collected sets of stems from different nurse sites and brought them back to the lab to dissect them to see how many eggs we could actually find. So what you're looking at here on the left is a set of 100 stems collected from the University of Waterloo. On those 100 stems, we found 316 eggs. So we were really happy with the efficacy with which we could collect those eggs. But what this graph on this slide also shows is the positions that were recorded of where those eggs were found on the stem. So at what height on the stem were those eggs found? And we found the vast majority of eggs in the lowest 1 meter of the stem. So what this tells us is that if we are going to transfer these agents by moving stems from one site to another, we don't necessarily need to transport the 4.5 tall meter Phragmites stems. We can just cut the lowest 1 meter, and this will be a much more efficient way of transferring almost all of the eggs. So in the summer of 2024, we conducted the first trial of these stem transfer releases. So this was conducted on the campus of Ontario Tech University. And on this campus, they have a long set of planters that for years have been inundated with invasive Phragmites. So we transferred 18 blocks of 75 collected stems from nurse sites into these planters. The stems were collected from 3 different nurse sites, and they were transferred into the Ontario Tech site using 3 different treatments based on when the stems were collected from their nurse sites, how they were stored over winter, and when they were placed into the recipient site at Ontario Tech University. So in treatment 1, the stems were collected from the nurse sites in the fall, shortly after the eggs would have been laid, and then they were transferred directly into Ontario Tech. So in those cases, the eggs would have overwintered on those cut stems in the Ontario Tech site. In treatment 2, the stems were collected in the fall, but then overwintered in sheds at the University of Toronto before being transferred into Ontario Tech University in the spring, shortly before the growth season. And then in the final treatment, those stems were collected in the spring, shortly before the growth season. So those eggs would have overwintered in their nurse site, and then they were moved directly into the Ontario Tech site. So this is what that set of planters looks like on Ontario Tech's campus when the Phragmites is grown up during the season. And we spent the summer monitoring those 18 different release plots to look for signs of damage from the biological control agents. And we found at all 18 of those plots substantial damage from the biological control agents. So this showed us straight away that not only can we reliably harvest eggs from nurse sites by collecting stems, but we can effectively transfer those populations into new Phragmites infestations just by setting up those cut stems in the new location. What we also found was no significant differences based on where the stems were collected from, so which nurse site they came from, and no significant differences based on those different treatments. So there's a lot of flexibility in terms of when the stems can be collected from nurse sites, where they can be stored over winter, and when they have to be transferred into the recipient site. So that flexibility can potentially go a long way in terms of allowing us to conduct more of these releases and also to oversee releases that might be conducted by other stakeholders. So in the summer of 2025, we conducted another experiment. This was a large experiment looking in part at the potential benefits of mowing patches before we release into them, to see whether that might increase the efficacy of some releases. But secondarily, what we were doing was comparing these stem transfer releases with our traditional egg and larval release techniques. What we found was that the stem transfer releases worked significantly better than our traditional egg releases. We had more damaged stems associated with these stem transfer releases than we did using the egg releases. And so these have now become not only an effective way of moving insects from nurse sites into new locations, but potentially our second most effective release method that we have at our disposal. So having developed this method now of moving insects from nurse sites into new locations, we're changing our tack somewhat in terms of how we use our lab-reared insects. So for example, rather than spreading ourselves thinly across the province, each year. What we did in 2025 was to conduct 5 very large releases in Durham region, with the hope that these releases will become nurse sites that can be used as sources to disperse insects throughout that Phragmites management area. And so we're now looking to identify and focus on a different Phragmites management area with each summer. So in the coming year, 2026, we have 8 sites planned for large releases of larvae and eggs in Essex region conservation area. And again, we'll continue to monitor those sites, and the ones that become the most effective nurse sites will be used to redistribute the insects throughout Essex region. And so on. During 2027 and 2028, we haven't identified these regions yet, but we're looking to identify Phragmites management areas in both the north and the east of the province that can again be used as source sites for dispersing the insects across the province. So a nice kind of announcement that we can kind of make at this point, having been monitoring these insects across 5 or 6 years, is that we're happy now to describe Arcanara neurica and Lanisa geminipuncta as established in Ontario. As I've focused on, we've developed this method of transferring insects from nurse sites into new locations, and that's going to be a huge tool by which we expand the program through this nurse site strategy across different Phragmites management areas. And so we're always very interested in talking to anyone who might be able to suggest Phragmites management areas or different release sites that we would be able to focus on. So I just want to finish with a quick thank you to many of our collaborators. Huge thank you to Ducks Unlimited Canada, Nature Conservancy Canada, and NSERC, and many of the conservation authorities that have done a huge amount of work and have helped us massively in this process. And so thank you very much to all of those, and I hope maybe I've got a couple of minutes to take questions.
Awesome. Thanks, Ian. That was a very interesting topic, interesting presentation. One of the things that stood out to me was kind of earlier on in the presentation, the distribution of the eggs along the stem. Really funny how the vast majority is under the first meter, and then there's like some overachievers at like 220 centimeters way up.
Yeah, there's a few. Yeah, it's interesting. I wonder whether they kind of, because they're not usually at the very low on the stem either. The bottom kind of 15 centimeters or so is usually fairly bare, and that's maybe a kind of an adaptation to avoid flooding. And maybe why they don't tend to go higher up on the stem is difficult to know, but it's possible some, in wind events, some of those Phragmites stems will snap halfway up. So maybe they're trying to avoid kind of the chances of that happening. But yeah, it's difficult, but it's a trend we've seen really consistently.
It's interesting. We do have quite a few questions for you here. How do you think this winter's very cold weather impacts your study?
Yeah, it's hard to know. I mean, what I would say so far, we don't know a huge amount about the kind of the extreme cold hardiness of these insects. And so, some research questions that we still have that remain to be answered are kind of how far north can we go in Ontario before potentially either the temperatures get too cold, or there might be a kind of phenological mismatch where the spring arrives too late. But I mean, what I'd say so far is that we've seen such consistent, successful overwintering of these eggs. They seem incredibly hardy. They're hardy both to extreme cold, and we've done experiments showing that they can survive submersion for several weeks underwater. And so they're a hardy bunch. I think my prediction would be that they'll be fine in this winter. But I do think this is probably our most extreme winter in terms of temperature that we've had so far. But quite a lot of snow cover as well. And so that may buffer the temperatures that those eggs are actually experiencing.
Yeah, very, very well insulated. Could the same insects be tested on Japanese knotweed?
Well, no. So these insects are highly, highly specific to Phragmites, and they're stem feeders. So they bore inside the Phragmites stems. So we do have biological control agents, or a biological control agent that is approved for release for Japanese knotweed. And it has been released over several years, but unfortunately, we're not seeing the same level of effectiveness from those Japanese knotweed. They're psyllids that we're releasing in that case. And what we think is happening there is that the nymphal stage of those psyllids is quite susceptible to both desiccation and predation, and those are our biggest worries about them. But we continue to monitor them, and we hope that eventually we'll start to see effects. But this Phragmites biological control program has huge potential, I think, beyond what we've seen with the Japanese knotweed agents.
Okay, awesome. Well, that's all the time that we have for questions. So if you want to stick around and answer some of the questions that are still in the Q&A box, there's quite a few there. So thank you so much for your presentation, Ian. Very interesting stuff.
Absolutely. Thanks, Alex.
And next up, we have Megan Ward from Trent University, and she'll be talking about lessons learned in invasive Phragmites australis, treatment, rolling leads to reduction in native vegetation diversity. So with that, I'll hand it over to Megan.
Hi, everyone. Can you see my screen okay?
Yep, you're good.
Okay, perfect. So thanks, Alex, for the introduction. My name is Megan Ward. I'm a PhD student at Trent University, and I study Phragmites invasion and management in Great Lake coastal wetlands. And today I want to focus a bit more specifically on a method of managing Phragmites called rolling that really hasn't been assessed in the scientific literature too much, and I want to share some of my findings with you all. So as we all know, wetlands, both inland and coastal, really benefit the economy. They support biodiversity and they protect infrastructure. And they do that in a variety of different ways. They can clean water, protect water supplies, they provide food and habitat, etc. So they are critically important habitat that we rely on quite a lot. Despite how important they are, we've seen more than 68% of wetlands being lost in Ontario since about the last 200 years or since European settlement. And this is much more stark in certain areas than it is in others, specifically in that sort of southwestern tip of Ontario there along the northern shore of Lake Erie. We've seen up to 100% of loss of wetlands in those regions. And that can be due to a variety of different reasons. There's a lot of different causes of wetland loss in Ontario, but what I sort of want to focus on today is land conversion and development and hydrological manipulations. And the reason I want to focus on those is because land conversion and development might lead to the draining of wetlands, and hydrological manipulations, for example, the construction of dams, may change water levels in wetlands, specifically in Great Lake coastal wetlands. And when you have both of those things happening, land conversion and draining, and hydrological manipulations and dams being created, we often see invasive species starting to take over. And it's because land conversion and hydrological manipulations are such large-scale disturbances that invasive species can come in and thrive in areas that are under a lot of pressure. And of course, the invasive species I'll be talking about today is Phragmites. We see Phragmites in every single Great Lake. It's present at multiple Great Lake coastal wetlands and has quite significant populations along the coast of Lake Erie and Lake Ontario. And when it does invade, it modifies the habitat in a variety of different ways. The first is through excess biomass production, and this is something a lot of us are probably familiar with. You see these intense growths of stems and leaves and shoots, and these biomass produces itself every single year and will have dead biomass remain standing year after year. So unlike a lot of species that will decay or decompose over the course of one season, Phragmites doesn't necessarily do that. And so we see this excess biomass production and retention over time. It also modifies the habitat through resource control. If you ever stood in a patch of Phragmites, you know how tall it is and how dense it is. So it can control things like light and nutrients and water in a way that reduces the fitness or the strength of other wetland plants that may be native. And finally, it has a lot of structural habitat changes. So when Phragmites invades, the root system can be so intense that it actually can start to terrestrialize wetlands. And you can kind of see that in this figure here on the screen, where you have this large, dense, thick mat of Phragmites upon which you can actually walk. And some folks, or in some locations, you can walk on these really dense Phragmites mats with only rain boots on, and you're not getting your socks wet. You don't have to wear waders, and you're not out in a canoe. And so this can functionally, quote unquote, terrestrialize a wetland. You have this wetland that used to have open water and changing water levels, all of a sudden to a wetland that is essentially terrestrialized with no standing water. And so, of course, there are a variety of methods that we can use to treat Phragmites. There are herbicide applications that have been very successful and arguably have some of the best efficacy. Lots of folks will pair herbicide with things like hand cutting, mowing, and burning, again, to sort of treat that idea of the above ground biomass that can have such negative effects on plants and animals. And then biocontrol, as we just recently heard from Ian. And so these three different sort of general treatment methods are pretty well researched, and we've seen positive results in the literature and in field applications coming out showing that these methods are working. What we haven't seen as much in the literature, and what not as many folks are actually doing for Phragmites management, is called rolling. And so rolling is when you cut the stems at ankle height or so and push over big thatches of Phragmites, like you can see in the photo on the right here, dense, thick mats of Phragmites being pushed over after the growing season. And I am specifically interested in assessing this method of treatment in non-inundated habitat, or habitat that doesn't have standing water. As I mentioned previously about habitat modification, Phragmites is terrestrializing a lot of different wetlands. And so previously, rolling used to be used when there was open water, when you could cut down dead material, perhaps after herbicide management, and allow the decomposers in the water to decompose the Phragmites over the course of 1 year, maybe 2. But because of this act of terrestrialization and reducing the amount of open water that we have in wetlands, a number of managers now are using rolling as a sole technique, but not able to inundate those plants, not able to have those plants decompose in water. And so our goal is to assess the method of rolling Phragmites in non-inundated habitat. And we ask this question: how does the treatment method of rolling Phragmites stems affect both Phragmites growing back in the next year and surrounding native plant diversity? And so this work took place at Point Pelee National Park. So Point Pelee National Park is Canada's smallest national park. It's on the northern shore of Lake Erie, but it's actually the southernmost tip of mainland Canada. And it's comprised over 70% of marshland habitat. And this marshland habitat is an endangered habitat, and it's of international significance. Despite sort of the international recognition of how important these wetlands are, we've seen significant invasion of the wetlands at Point Pelee through with Typha glauca, the hybrid cattail, and then also Phragmites. And so in the northern area of the marsh at Point Pelee National Park, they have a floating mat, which again has sort of terrestrialized these wetland habitats. So despite the fact that we're technically in a marsh within this area, my field techs and I can walk in this area and we're not seeing any water, we're not sinking down to our knees, and we're just wearing rain boots. So at Point Pelee, they tried to target 3 different patches of Phragmites growth using the rolling method. So they cut it at ankle height and rolled over the Phragmites. And so this produced a really interesting study design for us, because we could look at 3 sites of impact, 3 sites that had been rolled as a treatment method, and compare that to 3 sites in the north, in the red there, where no management had taken place. So those red patches are Phragmites patches. They're heavily invaded. You can even see on the Google image here, those like thick patches of Phragmites all along the north, and they weren't treated. So we had a really nice comparison of treated sites and untreated invaded sites. And this is what these untreated sites looked like before treatment. In 2020, we went in and collected stem density data, and we took measurement of all the different plants that we found. And these are some photos. So we can see that in these sites, Phragmites stem density is well over 100 stems per meter squared. So incredibly dense sites that were growing at Point Pelee. And then this is that same site after rolling. In May of 2023, 1 year after rolling, we can see that a lot of that Phragmites has been cleared away, almost like this open patch of land, and that there's a lot of dead material on the ground floor. In July of 2023, once things have started growing again, unfortunately, what we observed is just more Phragmites. There's a little bit of jewelweed in the corner there in that photo, but almost all the live plant material we saw growing in these areas that had been rolled was just more Phragmites. And so we really wanted to assess what happens when you roll, and is it a useful method if it's being used on its own in an area with not a lot of standing water or no standing water. So to do that, we compared native plant richness, so the number of different plant species in our plots, among the control sites in red and the treated sites in blue. So just as a reminder, these control sites are invaded with Phragmites. They've never undergone treatment, and the treated sites were rolled. In 2022, this took place before any method of treatment. So this is our before treatment data. And we can see that native plant richness is pretty comparable between these treated and control sites. On average, we have about three to four species of native plants per plot when both sites are invaded by Phragmites. In 2023, we actually see that blue value decline. And so what that tells us is that 1 year after treatment in the treated site, there were actually less native plants than there were before treatment. And there are less native plants in the treated site compared to the control in 2023. So this is quite a negative finding. This tells us that rolling as a treatment method actually reduces native plant richness after 1 year. And unfortunately, we found the same trend, but just continued in 2024. So 2 years after treatment, we're seeing a sustained decline in native plant richness, but no change in native plant richness in our control sites. And so the general takeaway from this figure is that if you're rolling Phragmites in dry sites, you're actually reducing native plant richness. You're making it harder for native plants to survive. So this is not the most positive of findings, but useful when we're trying to inform ongoing Phragmites management. We also then looked at live Phragmites stem density, so the number of live individual stems per meter squared. And we compared that between our control and our treated sites in 2022, 23, and 24. There was no significant difference in the number of stems in 2022 among these different sites before treatment. And while the number of live stems did decrease in 2023 in our treated sites, so in those blue dots, what we were hoping to see was 0 blue dots, or blue dots all along that 0 stem line, because what this is telling us is that even though these patches of Phragmites just underwent treatment the year before, we're still seeing more live growth the next year. And unfortunately, that number of stems that are alive, that are growing every year, increased in 2024. So this tells us that not only did rolling not impede growth in the first year after treatment, but in fact, in the second year after treatment, we're seeing even more live plants continuing to grow. So again, not a useful finding when we're hoping to see a complete reduction or at least sustained decline in the number of live stems growing. We also looked at live and dead stem growth within these individual plots, so both live and dead combined. And there was no difference in 2022 among these sites, which is useful as sort of like a baseline comparison. In 2023, that number shot way down, again, because all of the live and dead plants in 2022 would have been pushed over. And so what we're seeing here is only live plants in 2023. Unfortunately, in 2024, that value is increasing. And so this tells us that, like the previous graph, not only are we seeing more live plants, but also all those dead plants from the previous year are still remaining standing. So we still have this issue of a lot of dead biomass being collected every single year after the rolling treatment. And so we have sort of a main takeaway from this research. Rolling Phragmites in non-inundated wetlands or wetlands that don't have any standing water ultimately fails. The Phragmites returns quickly within 1 year, and native richness declines and has a sustained decline. There is no lasting reduction in Phragmites stem density. There's native plant richness declines post-treatment. This is a really important part of the project because not only in Phragmites management are we looking to see if the Phragmites itself goes away or is eliminated, but in response to that, we want to see native plants coming in and taking over that area again. And so with this treatment method, rolling in dry sites, not only do we see a return of Phragmites, but actually a sustained decline in native plants. Our sort of general takeaway is that rolling alone is not a viable restoration tool if you don't have any type of hydrological intervention to, for example, increase flooding or any follow-up treatments. Rolling with herbicide application in inundated sites, in sites with standing water, might very well be a viable application, but on its own, Phragmites is unfortunately not controlled if rolling is the only method applied. And so with that, I had a whole whack of field support to help me collect all this data over the course of the 3 years, and a quick thanks to our funding partners as well. And if anyone has questions, I'm happy to chat about them now. I can answer them in the Q&A box if we don't have the time, but also feel free to email me if you want to chat a bit more, or if you would like the early version of this manuscript that's currently in review. But I'm happy to take questions. Thank you.
Awesome. Thank you, Nathan. That was very interesting. There are a few questions I'll read out to you. One thing I wanted to note before I get into that was I noticed an interesting, I suppose, natural parallel. Here in the Sioux, we had a lot of snow last year, and this year I think it's just kind of becoming the norm that we get a lot of snow. And the areas that leveled the frag, I noticed, came back very strong the next year. So I wonder if the areas with kind of the lake effect snow or heavy snowfall kind of get that helping hand each winter.
Yeah, perhaps.
Was the rolling treatment only completed once, or was it repeated annually during the study?
So it was only completed that one time in the fall of 2022. So we collected all of our data in July. In September, October, the rolling took place, and then we collected data the following 2 years, but rolling itself took place once.
Was the rolling method attempted alongside any other treatments?
No, so in this research, rolling is a standalone method. I do think rolling, in addition to other treatment methods, might very well be a viable option, but rolling in and of itself in this project didn't work. But no, there were no other treatment methods.
Yeah, I've heard of it being used to make it so that when this new sums come up, they're more easily accessible for management.
For sure, yeah.
What type of equipment is used to roll?
So these guys use like raspberry cutters and like essentially weed whackers to just sort of push it over. You could use individual cutting if that was of interest. And if you're dealing with anything other underwater, the materials might be different. But these folks at Peely then just used like weed whackers and raspberry cutters.
Okay. Is there any new management strategies being applied to the same site?
Not at this site, particularly these 3 individual patches. Point Peely National Park is struggling with 2 significant invasive species, the hybrid cattail, Typha glauca, and Phragmites. And so right now their movement has moved a bit more towards the Typha glauca, and they're creating a lot of hydrological channels and changing the hydrology of the landscape. And so that's where their focus is with their methods sort of moving forward. So I don't anticipate any follow-up treatment at these sites.
That's all the questions we have for now. Some may pop up. Oh, one just popped up. We do have a quick minute. So do you think the native richness result would be different when the treated area is smaller or closer to the source of native plants?
I think the native richness results would be different if the dense layers of thatch, like dead material that were left behind after rolling, were removed. What we anticipate happened is that 100% of light penetration was impeded. The microclimates changed. It was probably really hot underneath these dense, thick materials of dead material. For example, some of these piles of dead Phragmites that had been laid down were almost a meter thick. So we could like barely walk through them. So native plants trying to penetrate through that isn't realistic. Perhaps if there was a really significant native seed source, like in the seed bank, this site is actually quite close to other emergent vegetation that is growing, so it is close to a native seed source. I think the sort of mechanism that caused that decline was the accumulation of the dead thatch, the dead leftover Phragmites.
Well, thank you very much, Megan, for your presentation. It was very, very interesting stuff.
Yeah, thanks for having me.
And next up, we have Michael McTavish from Rare Charitable Research Reserve. He'll be talking about the morphological identification of native and introduced Phragmites australis. So with that, I'll pass it over to Mike.
Perfect. Thanks very much, Alex. I'll pull up my screen here. That coming through okay? Okay. Perfect. Okay. So good morning, everyone. Very happy to be here. So I'm the conservation scientist at Rare, and I'm going to be chatting about a piece of older work that we're continuing to develop going forwards that has to do with more of the identification side of the Phragmites challenge. So as I said, this is a slightly older piece of work that actually started, the core piece of work was during my postdoc out of University of Toronto. So this was working with Sandy Smith and Rob Boucher, Tyler Smith and Subaya Machanda out of University of Toronto, Forestry and Agriculture and Agri-Food Canada. Ian Jones, who we heard from as well, was also contributed to the development of this. And this is continuing as a part of a project that we're doing out of Rare now as well. For those not familiar, briefly, Rare is a community-driven land trust and an environmental research institute. We're based in Cambridge, and we work on the interacting pillars of conservation research and education. And I run our research program there. We're all familiar, I think, within this session with the challenges of Phragmites as one of the worst weeds in North America. And an important part of management is being able to effectively identify the target weed that we're dealing with. And with Phragmites, this is important because, of course, we have multiple lineages that we might encounter when we're out in the field. So we have introduced Phragmites, Phragmites australis, which on the one hand, again, we talk about as Canada's worst weed. It's very common and spreading and very damaging, as we know. But on the other hand, we have the native subspecies, which has been alluded to, Phragmites australis americanus. In contrast, this is a desirable native species. It tends to be much rarer, although that does depend where you are geographically within the province, and also tends to be on the decline, in part from the effects of its introduced counterpart. And so being able to tell these apart is really important for targeting management. And there are some very strong identification resources. Some are better than others. Many, however, primarily rely on these kind of general rules of thumb, I would almost describe them. So, for example, native Phragmites is generally going to be smaller. It occurs at lower density. It has these smooth red stems, whereas introduced Phragmites then is larger, occurs at high density and has rough brown stems. And that is often the case. But anecdotally, we also do run into a lot of situations where people are encountering what they would describe as these atypical stands. So let's say you run into something that is kind of moderate density, it's maybe medium stature, there's some red in the stems, but not fully. What are we actually looking at there, and how do we try and figure that out? Now, fortunately, we do have some wonderful tools available to people. We've already heard about the Ontario Phragmites Action Program's genetic testing program, which is really one of the best tools that you can use here, and I would encourage people to check that out if you're interested. But there remains additional interest in complementary tools that we can implement easily alongside that genetic testing. And so what we were really trying to achieve here from a management perspective was to produce a free, easy to use identification resource for Phragmites that land managers and researchers could use to complement other tools like the genetic testing. From a research perspective, what into is trying to identify some of the best morphological traits that can distinguish between these subspecies that are reliable for identification, are also quick and relatively easy to use. And so the actual fieldwork from this goes back a ways. This is actually from fall of 2019. This was developed in the very early stages of my work with the biocontrol team that we just heard from Ian. As we were developing some of those initial biocontrol tools, we were also working on this identification piece. So one of the first things that was actually done was a field survey and collection of 21 introduced and 27 native Phragmites populations across southern and eastern Ontario, as you can see on the map here. At each location, there were 5 subsamples collected. A few measurements were taken in the field, but most of the measurements were actually from the lab. So I was harvesting stems, taking those back for measurement in the lab. And dried leaf samples were also collected so that our Agriculture Agri-Food Canada colleagues could provide genetic identification to clarify exactly what we were looking at in each instance. We have here a suite of 22 morphological measurements that were determined for each of the samples, with details about how they were calculated, why we were interested in looking at them, how this was parameterized, and so on. And essentially, we were looking at all different aspects of the plant, stand level characteristics, the stems, the leaves, the florets and spikelets, the panicles, really all over the plant. At the core of this, we're trying to ask, first of all, well, what is the mean and range of trait values for each subspecies on average? And then do those differ, basically, between those populations? But simply having a difference between the two is not enough from a diagnostic perspective. Ideally, we want traits that are fully separable and have no overlap between those populations. So we're trying to compare that as well. So there's a lot of information here, but in essence, what we found is that basically all of the traits differed, and mostly in the expected way between subspecies. So things like size and coloration, they all played out the way that we expected. But what we found is that these traits were also highly variable, and most had substantial overlap between the subspecies as well. So there were measurements that could say this was a native plant or this was an introduced plant. We can look at this graphically here. There's a lot going on, but essentially each of these dots is an average trait value that's been normalized on a scale from 0 to 1. Blue dots represent averages for introduced populations, open white circles for native populations. And as you can see, there's a lot of that common ground in the middle where those trait measurements actually have quite a bit of overlap. In fact, there are only 3 traits that we measured that provided complete separation. And we'll look at these in more detail later, but these were the percent of old leaves that were retained on dead senesced stems, the coloration of the basal section of the stem, and the height of the ligule. We also found that there was one particular measurement, which was the percentage of stems that had this spot fungus occurring on it, only occurred on native species. It wasn't always there, so its absence is uninformative. But if that spot fungus was present, that was diagnostic, in our samples at least, for the native subspecies. This one was a little bit more complicated, but from some of Tyler's work in particular, we were able to identify a sort of composite trait that if you combine leaf length as a measurement and the length of the lower glume, you arrive at sort of a simple binomial key that will also provide complete separation between the subspecies as well. And so these 5 traits or composite traits were put together into a sort of ID checklist guide as shown here. And you can access this and the table and all the information we're talking about today in an open access article in Invasive Plant Science and Management. So I'd encourage you to check that out if you're interested. This guide includes a how to measure sort of description in the table itself, as well as more detail within the paper. You essentially measure a given stem one at a time, and then check off the corresponding box based on your measurement, and that'll align with either the introduced or native side of the table. And ideally, we're looking for complete consensus between all of these checkboxes. It all points to the same subspecies. If there's measurement error or if the population lies beyond what we were able to assess in our study here, then that would suggest further genetic testing is a required step at that point. So to give a bit of a closer look at some of those measurements, this one was really a standout for me, and the one that I share most widely with people asking about this really is leaf retention. So introduced Phragmites, when the stems die, a lot of them remain standing on the landscape. And the leaf sheaths that are curled up against the stem tend to be retained in very high numbers, whereas native Phragmites, the vast majorities of those are shed off over time. And this is a really nice trait because it's easy to see, and it tends to persist on the landscape for multiple years, and you can see it throughout the seasons. So really, really good trait that I would encourage you to watch for. This shows up a lot as well, the ligule, so this membranous tissue right where the leaf meets the stem. If you exclude the sort of hairy variable portion at the top and just focus on that dark colored band, if you measure that height right in the middle, that provided good separation as well. I mentioned that stem spot fungus, which when present points towards the native Phragmites. When absent, that still could be either subspecies, so that's not diagnostic. And similarly, stem coloration as well. We see ability in this trait, but in our samples, that very deep dark red that covers basically 100% of those lower internode segments of the stem really only occurred on native Phragmites. So again, diagnostic when it's there, we don't know when it's absent. And then here's that composite trait as well. That's a combined measurement of the lower glume and leaf length. This one is a little bit fiddlier just because it is this two-step process. Also, measuring that glume length ideally requires taking some of those spikelets and looking at them under a microscope. So maybe not as accessible as the other traits there, but doable with a little bit of equipment as well. So just a few quick things to keep in mind if you're looking at using this identification key. We sampled 48 populations. These are from Southern Ontario. There's a lot of frag, of course, in the province. And so we tried to make this as generalizable as possible, but it is from this particular region and these 48 populations. So keep that in mind, especially if you're trying to apply it beyond that region. These measurements were collected in the late summer and the fall, and so for best comparability, that would be the time to take those measurements as well. Some of the traits are going to be more seasonal than others. I noted the leaf sheath retention is particularly nice because as long as you're able to find some older stems that are there, this is effectively a year-round trait that you can look at anytime. I noted that some of these trait results are not going to be diagnostic, so if you don't see that stem spot fungus or you don't see that deep dark red, that's not actually providing any information in that case. The ID guide is intended to be used on a stem-by-stem basis, but you should be checking as many stems as possible from representative locations throughout a patch to have a higher confidence in what it's telling you. I'm going to expand on this in a moment, but the test populations for the study did not include hybrids, which we do now know occur in Ontario. And so again, more on that in a moment. And overall, well, we think this is a complementary tool and something that can be applied very, very quickly in the field. Remember that, of course, that ultimate answer really does come from genetic testing. So I would encourage you to check out the OPA program for that as well. So a few quick notes on the ongoing nature of this research and some things we can take away from this. The big one, of course, is that we do have hybrid populations in Ontario, as Darissa was mentioning earlier. So a really important thing going forward will be to collect some measurements from some of those populations, and that's something we hope to do later this summer and fall, and try and assess how morphologically distinctive native and introduced frag are alongside these hybrids. So what does that mean for this kind of morphological identification? To that point, we're also working on an updated Phragmites identification resource alongside the Invasive Species Center. The first pass at this may or may not include that hybrid information, depending on the timing, but we'll probably go through a few rounds of this. One thing we're looking to do is to incorporate some additional validation of what we're seeing in the guide using additional data that has been collected through that genetic testing program that we've mentioned. And so finally, a few takeaways here. Effective management of Phragmites requires reliable identification of which subspecies we are looking at. Native and introduced plants are different morphologically, but there's also a lot of variation in those traits and significant overlap. And for that reason, what might be considered an atypical trait is actually often quite normal and within the normal range of variation that we know exists. And for that reason, these kind of rules of thumb, invasive frag being big, native frag being small, and so on, are really insufficient and can be misleading. So what we really require are these data-driven diagnostic traits and ongoing assessment of that as well across a broad geographic range, and especially now with consideration of hybrids and what those look like on this identification spectrum. So again, I would encourage you to check out that paper that our old group did. It's available open access, and keep an eye open for future invasive species centered partnered guide work as well with some of those updates. So thanks very much, and happy to take some questions.
Awesome. Thanks, Mike. Also very interesting stuff. I appreciate all of the DNA program plugs. We do have a few questions here for you. One of them you kind of answered is the overlap in traits due to hybridization?
Yeah, so that their hybridization occurs much less often than it does happen, but it is relatively rare in the field. In the US, we have a few more example populations where they've been able to look at it a little more closely. For us, it's still so new. I know from the US, some of the work does suggest sort of intermediate morphological traits. So you have intermediate genetics and intermediate traits, essentially. And that's kind of the concern here, is that it would potentially muddy the water of identifying what we're looking at. So it might be a case of winnowing down what is truly diagnostic now that we have this sort of third option in contention as well. So that's something we're going to continue to evaluate as more hybrids potentially show up.
Right. And I'm excited to work with you on that.
For sure.
Are you aware of any genetic testing available in the States?
I don't have specific context for that, but I'm sure that I would be very surprised if there wasn't work being done somewhere. Others might be better informed about that, but I'd be surprised if there weren't options. They also have the Gulf Coast lineage as well, so they have an additional one in the mix that we don't need to be concerned about up here as much.
Okay. I do believe I have a contact that does do genetic testing in the States. So if you want to reach out to me, I can get that to you. I don't have it right offhand, but yeah, if you reach out to me, I can hopefully get you that contact. How common do you encounter native and invasive for other hybrids?
Yeah, so very rare. Really, we have sort of the two pockets, I believe, in Ontario so far, as mentioned, on Georgian Bay and then up near Sudbury. But as always with these sort of things, it's a function of our awareness of this as an option and how carefully we look as well. So, but again, from the US, it is still relatively rare overall, which is encouraging.
Yeah, and hoping to get a better idea of the proper demographic distribution of the hybrid as the genetic testing program continues and we get a better sample area.
Certainly.
Is RARE part of a PMA working group?
Yes, so we are RARE is a part of the relatively newly formed Waterloo Region Phragmites working group management area. We're actually helping to extend the biocontrol program that Ian talked about that's based out of University of Toronto within that PMA specifically. So using that nurse strategy that Ian was talking about and with support from their team, providing a bit of local on the ground distribution and sort of support for people looking to use that tool in that area.
Few people would like to know if there's native Phragmites in the GTA or Southern Ontario in general.
Definitely in Southern Ontario. The GTA, I'm trying to remember if we actually found some there. The further south you go, they do tend to be a little bit more sparse, less frequent and smaller stature, smaller populations overall. As you go further north, as was mentioned earlier, it definitely does become more common. There's that geographic patterning to it. It is present, but especially in areas where that introduced Phragmites is much more common, those competitive interactions between the two tend to be the native Phragmites fares quite poorly in that interaction. So that can really lead to either competitive displacement or, as we've seen, that potential hybridization as well. So it is around, but it depends where you are as well.
Okay. Oh, and someone put the Great Lakes Phragmites Collaborative in the US. They're the genetic testers for the United States. Okay. I believe that's all we have time for. So thank you again, Mike. There's a lot of good information. And yeah, I'll pass it back over to Maddie to give some closing words. And thank you all for coming.
Thanks.
Thanks so much, Alex, and thank you to all of our speakers for their presentations. That was a really informative session. We have several resources specific to preventing and managing invasive Phragmites, so if you're interested in learning more, you can check out OntarioPhragmitesAction.ca. We'll take a 10-minute break now and rejoin at 10:40 for our aquatic plant session, so hope to see you all there again. Thanks.